I'm really going to try and do is to see how generic the properties of what are commonly known as dietary fibre polysaccharides. In essence, they're the components from the cell walls of the plant foods that we eat in various forms, and try and see how generic their properties are, linked by their physical characteristics more so than their chemical structures. So we think that it's the physical characteristics that really determine the behavior in the digestive tract and the consequent nutritional benefits. So that's really the thesis, and we're going to explore that as we go forward. So this is what you should be eating. These are the various dietary guideline cartoons from different jurisdictions. The one on the left is the Australian one, very complex, and you can't really see much of that. Through to the right, the top right is the US one, which is the simplest. No knife in the US, everything's eaten with a fork apparently. And that really captures it. I mean, the simplicity can be a bit difficult, but in this area it's not a bad thing to do. So it gives you an impression of the balance, not just on a single plate, but across the diet. And there is a consensus, basically, around what should be the constituents of a nutritious diet, and it's around that balance between whole foods on one hand and the different macronutrients they contain on the other hand, diversity being important, and really an emphasis these days on foods which are closer to the natural form and less of an emphasis on the molecular composition. So we're moving away from an ingredient-based dietary guidelines to more of a whole food-based dietary guidelines, which is appropriate in my view. So bearing those in mind, often we wanted to say, how do we get to this? How do we get to these dietary guidelines? And what are we doing? How are we travelling? What are the deficiencies that most people have in their current diets? But the other thing we also always got to remember, now on the left again, is the previous version of the Australian Dietary Guidelines, which in my view is probably better, as it's got this concept that, of course, it's diet and lifestyle, as exemplified here by the feet at the bottom. So there is this balance. Of course, there's variation in terms of what a healthy diet is for an individual based on what their lifestyle is, not just their physical activity, but all the other aspects of their lifestyle. So it will not be prescriptive. It will be something with broad outlines, and that's really the nature of the nutritional guidelines that various countries promote. Where it all comes from, though, is almost entirely from epidemiology. That is the science of food diaries and medical outcomes, effectively. Very simple, but gets more and more powerful as the food diaries are kept for longer and longer and longer, and the medical consequences get more and more, have more and more years to play themselves out. So at the moment there are at least 1 million, probably more than that, people around the world who are engaged in these ongoing epidemiological studies. And as time goes by, more and more of that data becomes available. Of course, our ability to interrogate them computationally gets better, and so we can get a lot of information around correlations. What is the correlation between people's intakes and people's health outcomes? But it's correlation, clearly, because there's no indication there of what causes something. It just says that this if you eat this, then the statistical chances are that this will happen nutritionally. Second most important point, and really the motivation for this whole area, is that really almost across the world, the area, the part of our diet which is underrepresented is the one which is rich in plant cell walls, plant tissues. So it's the whole foods, so there's no problem in getting people to drink juices, but to eat whole fruits, whole vegetables, whole grains, still a challenge. So there's, by and large, most people are not eating in the balance of their diet, are not having enough of the whole food, particularly vegetables and whole grains. And the risks of developing the major lifestyle diseases, diabetes, obesity, various cardiovascular diseases, colon cancers, and so on, are clearly associated statistically with, or negatively, with the intake of plant tissue foods. But the challenge scientifically is that this is all based on correlation, so we really need causation. So with the motivation for this work is really to say, that the correlations are out there, but the causations are not yet understood, so we're looking for mechanism. So this is what I'm going to talk about today. So a little bit about the translation from epidemiology towards mechanism, the way in which we're thinking around the generic physical properties and walking you through examples of those three areas of the roles of cell wall materials, dietary fibre materials in binding components in the digestive tract, in providing transport barriers to movement of materials within the digestive tract, and to the bulk structuring of the physical rheological properties of the digester. And finally saying, well, if we take all that in its total, can we go beyond the rather simplistic measures we have at the moment of dietary fibre, with the only categorization is soluble versus insoluble, which we believe is too limiting and we have the potential to be a lot more prescriptive in terms of the types of fibre if we are have better knowledge of the underlying physical properties that dictate the nutritional values. So if you go from epidemiology to mechanism, you think about the health challenges that we're interested in: obesity, diabetes, CVD, colon cancer are the ones that probably have the strongest relationship with plant food intake. And you say, well, what are the risk factors, which again are rather reasonably well understood, some of them. This is oversimplistic, but just to give you the idea. Sort of risk factors of total energy intake or imbalance of energy intake for obesity. Diabetes, a risk factor is numerous blood glucose excursions, so too much glycemic response over a prolonged period of time. Cardiovascular diseases, cholesterol and lipid levels in blood are risk biomarkers. And colon cancer, a low-fibre diet is a well-recognised risk biomarker there. So we can say, well, you can take those health challenges and say, well, here's some of the risks. So what are the roles for plant foods and dietary fibre effects in there? And so these are the sorts of things you can start to think about the roles within diets. So in terms of energy intake, a lot of that is driven by our lack of a stop signal in terms of eating. You know, we are driven to eat as prolifically as we can. That's our evolutionary heritage. But satiety is that feeling that stops us from doing that. And dietary fibre cell walls have several mechanisms by which they induce satiety. Diabetes is around now the roles of cell wall structures in smoothing out that glucose curve, and so preventing the spikes and troughs, the hypoglycemic troughs, and providing a mechanism of slowing down the digestion of starches and thereby, again, smoothing out the liberation of energy into the body. So the cardiovascular disease, again, there's probably the most well-known effect because there's a mandated health claim, at least with some jurisdictions, around some of the soluble fibres that they can reduce the cholesterol level in the circulation. Lesser known, but they can also reduce the total circulating lipids as well. And the mechanism by which this is proposed to happen is that it is proposed that these soluble polymers can scour out the bile salts from our system, and that the body recruits cholesterol from the blood to replenish those bile salts. That's the current mechanism. Colon cancer is something which is still a very big problem, at least in Australia and in many other developed countries. And really, a lot of that is probably going to be traced to the lack of fermentation throughout the colon. So fermentation is what happens to the dietary fibre when it gets to the end of our digestive tract. If that fermentation is either insufficient or does not persist to the end of towards the rectum, then that is when the risk factors for colon cancer come up. And colon cancer is most prolific at the rectal end of the colon. So promoting fermentation throughout the colon is a very important thing to do. So we can hypothesize various mechanisms by which cell wall dietary fibre polymers can affect satiety by gastric distension, or they can affect it by hormonal feedback from fermentation in the colon, and so on and so on. So there's a number, you can build a logic here. So we looked at all of this and we said, well, okay, well, how do we then take that to something which allows us to predict the properties based on the structures that we create in our foods? And for that, we're really interested in both the rate and therefore the site of digestion. So when we are eating something, we have a certain time scale. So food naturally passes through our digestive systems at a certain rate. It does vary. Meal to meal it varies. From individual to individual it varies. But the kinetics are the driver here, because we have a certain time that our food is in different parts of our digestive tract. And so the rate at which something happens also determines the site at which it happens. And that becomes an important thing when we start talking about what gets to the large intestine or survives from the small intestine. We'll come back to that later. So we have the various stages in the digestive process, of course, with the chewing, swallowing, holding in the stomach. The stomach is a holding tank as a way of metering out food into the rest of the digestive tract, then through into the small intestine, which has a very high surface area, which is very efficient for absorption, and then and if you survive the small intestine, you enter the large intestine, which has a less of a surface area but a much wider vessel, and that is where our microorganisms primarily reside. So we can start thinking around factors around what controls the rate at which things are emptied from the stomach, what controls the site of digestion, because there are these phenomena called the ileal or colonic breaks, which are hormonal signals, basically saying, I don't need to eat anymore because my digestive tract is full. So if we get signals at the end of the digestive tract that prevent us from eating, or from either releasing from the stomach or from eating more. And as we've talked about, there are factors involved in the glycemic response and the amount of starch that escapes digestion. And also we'll come back to, it's also about the micronutrients, not just about the macronutrients. Plant cell walls or dietary fibres are essentially the way in which our micronutrients are passaged through our digestive tract. And so the way where they are released has a big effect on the consequences in terms of micronutrient delivery as well as macronutrients. So we're actually remarkably naive of what happens in the human small intestine. It's very difficult to get to, clearly. It's difficult to get to from the top, it's difficult to get to from the bottom. It's the most inaccessible part of our digestive tract. But the sort of things that go on that we need to take account of are the entry of materials at the top of the small intestine in the duodenum, where we have bile as the emulsifier and various enzymes. Then as we go through the small intestine, then the products of enzyme digestion, glucose from starch, amino acids from proteins, fatty acids from triglycerides, and a range of released micronutrients are available for absorption. And as we go through, also water is taken up, salts are taken up, and at the end of the small intestine, the bile is recycled so that we don't lose it and we can recover it and take it forward. So part of our active research is really understanding how this breakdown process occurs, and the blue pieces here are the digestible parts of the food, the red pieces are the plant cell wall materials, and the critical factor is we do not have in our small intestine, we do not have enzymes that break down plant cell walls. And that's, if you take home only one message, that's it. It's very simple, but that really is the heart of how they have these big effects in our digestive tract, because all the other major components of food are digested in the small intestine. So it is the carrier, it is the modulator, it is the physical structuring, and as that goes through, as you get less and less of the other digester, of course, the remaining undigested material becomes more and more dominated by the fibre components. So what gets the large intestine is a really important thing, because that's what feeds the gut microflora. But what gets to the large intestine is a negative thing. It's what hasn't been absorbed. So it's not something you can measure in a food before you start. It's not predictable because it depends on the time and the amount of enzyme which people secrete, and therefore is not something which is uniquely to do with the food. It's also to do with the person. So this is a difficulty in this area, that what gets to the colon cannot be defined precisely based on the food, unless you're talking about the plant cell walls, because we know that they will all get to the large intestine because they cannot be broken down. But how much protein, how much starch, you cannot predict from the food. Well, you can have an estimate, but you cannot be precise about it because it's a negative thing. And what gets to the large intestine then becomes the feedstock for the microflora, the gut microbiome, which is that area of science which is just exploding at the moment in terms of its impact on our general bodily health. I'm not going to go into details. There are other people who are better placed to do that, but just to show you the type of early days we are basically in this field. papers in Nature from a couple of years ago, really very simple in a way, but very informative. They were comparing here what happens to the what the microbiome, that is the sum total of all of the microbiota in the large intestine, how that changes for a cohort of non-obese and obese subjects. And the key finding was it's really about complexity. So non-obese subjects have a very complex, diverse set of microflora, whereas obesity tends to be associated with a narrowing of that diversity. The good news, and that was associated also with the metabolic disease risk. So we know obesity is a big risk factor for diabetes and cardiovascular disease, but it seems that much of that could well be mediated through the microflora. It's possible. It's not yet proven, but that's where people are thinking there's a good chance that is the case. The good news, though, is that the worse your complexity is, the easier it is to respond to dietary intervention. So that's the really important thing from our point of view, that these are factors which can respond to dietary intervention, and it can be quick. It will take of the order of 3 to 5 to 7 days to really quite significantly change your microbiota, because the bacteria, which are primarily bacteria, that have such short generation times that it doesn't take very many generations for a cohort of bacteria to evolve that are better suited to the health of the host. So we really can influence the health of ourselves, the host, if we like, of our microbiota through them and in a quick time, because of the generation time of bacteria being on the hours time scale, so that therefore we have a lot of chance to really manipulate this. And watch this space. This is an extremely active area of research at the moment, and we need to learn a lot more about it. So, summarizing what happens in our digestive tract, one important point to say is the mouth. If we're taking in solid food, clearly, obviously, we chew it and we reduce the particle size. But in terms of plant foods, that's the last bit of structural breakdown that occurs until we reach the large intestine, because most plant foods are really not that greatly affected by the processes in the stomach and the small intestine in terms of their plant cell, in terms of the cell walls themselves. In the stomach, though, depending on the size of the particles, the degree of swelling, this is where the satiety effects kick in, and they prolong the residence time. So prolonging residence time in the stomach is a good thing. So if you have your porridge or your muesli in the morning and it's still sitting in your stomach 4 hours later, that's because of the physical properties. It's still there because the stomach is still processing it and allowing bits of it through all the time, which is a very good thing in terms of metering out your response to macronutrients. And then, of course, we go into the small intestine where we have the enzymic digestion of the major macronutrients, proteins, fats, and starches, but not the cell walls. And here's where the barrier and structuring effects really come into play. So if your macronutrients are encased in a plant cell wall, that's a very different circumstance to if they're floating free in the digester. Has a really a major effect on the digestion rates. So we've got to think about this is really the heart of the intact foods versus blended or comminuted foods, that we're preserving the natural encapsulation, and as we'll see, that has a big effect. And as we've talked about already, that when you move to the large intestine, then a lot of fermentation goes on because the dietary fibres are the major carbon source for the colonic bacteria. Importantly, though, we want to maintain that fermentation throughout the digestive tract, throughout the large intestine, and so we also want to know the rate of fermentation because that determines the site of fermentation. And anything which is trapped or bound up in the small intestine within cell walls that cannot get degraded there are now released because our microbiota do have the ability to break down the plant cell walls and release whatever's inside them. So we're talking about these sorts of structures. So we're talking about cell-based structures, and the cells are of the order of 100 microns. I mean, obviously it varies quite a lot from plant to plant, but of that order. But most of the pieces, we have whole pieces, we're eating much bigger than cells. So we're eating things which are obviously multicellular tissue-like. But when we break things down, we can break them down and release cells. We can also break them down and open the cell walls, and we have polymers that are released and so on. So we have all sorts of structures from the millimeter down to the nanometer scale, and we have to account for all of those as we go through our digestive process. So, plant cell walls meets gastrointestinal tract physiology. What happens? Or what are the things that we need to think about? So this is where we categorize the factors. This is based basically on a large number of diverse number of studies, but there seem to be commonalities in them, and we think we can just look at the underlying properties in terms of these three parameters of binding, of transport, and structuring. So that's what I'm going to talk about in the next section of the talk. And it's really about how those three properties of dietary fibre cell wall materials meet the physiology, the anatomy of our gut, the biochemistry or the enzymology that's going on in there, and then the microbiology and fermentation processes as well. So binding is probably more widespread than we had initially anticipated. So plant cell wall materials are very good at binding things. And we'll see they can bind bacteria, they can bind enzymes, they can bind nutrients, they can bind bile salts. And you've got anyone that's wearing cotton, you know what happens when you spill fruit juice or you spill red wine or coffee on that. Things stick. And this cotton is cellulose. So things stick. Cellulose is a really good substructure onto which things stick. And it's not just things like coffee and wine, but it's also bacteria, enzymes, all sorts of things. So this is a fairly, I hope you can see some of that. This is just the idea that what binds to cellulose in this case also determines which bacteria survive or can prosper based on using the sugar from that cellulose. So the red in here is cellulose. The bright yellow are the bacteria which are associated with that cellulose, and the green is the other general bacteria. So there's a subset of the bacteria which are able to bind to the cellulose, and that's important for them because they can then feed off that cellulose. Cellulose is just a polymer of glucose, and they can break it down into glucose, and that is their feed source. And so that will really start to be one of the factors that determines this relationship between the nature of the cell walls and the bacterial community that is derived, and the nature of those, the microbial components of that community. The sum total of that's our microbiome, and that's what we're trying to link through to those big-scale health effects that we talked about earlier. In terms of enzymes, so we are finding that essentially any enzyme has a binding to plant cell walls. The strength and the dissociation or the off rate and the equilibrium constant will change a little bit, but some of them are really very strong. So if you take alpha amylase, the starch digesting enzyme, and you expose that to either cellulose or to wheat bran before it's seen any starch, and then you add starch into the system, the starch is untouched. So the amylase just sits on the bran or the cellulose. Now, this isn't what happens in vivo. The amylase gets squirted into the whole mass of digester. But if there's enough bran or cellulose in there, quite a lot of that amylase will get stuck onto that cellulose. And we think this is a very interesting new perspective on how we may control the rates of glycemic response through the interruption of essentially insoluble cell walls. It's not just alpha amylase that binds. We've found various proteases will bind quite strongly as well. So there's something we're looking into here in a more generic sense that needs more work. In terms of nutrients, this is a bit more advanced. So we find a range of sort of classic phytochemicals are all bound extensively to cellulose. And the predicted capacities are large, so more than a gram per gram of cellulose. You tend not to reach those capacities because of the limited solubilities of these materials. But what happens? You have, in a plant, you have all these phytochemicals inside the plant structure. We break open the plant. They have a chance to bind, and they do. The extent to which they bind will depend on the time that you give for contact. It's the same thing if you spill coffee or wine on your cotton shirt, if you go and wash it off straight away, it's okay. If you leave it to wait for half an hour, no chance, it's stuck. And the same thing here with foods, that's what happens. So you create foods, you create those combinations of micronutrients and cellulosic structures that can bind. If you give them enough time and you give them, and there is enough and the ratio is appropriate, so I'm trying to say. So the ratio of cellulose to micronutrient is appropriate. You can get very extensive binding. I don't know how much you can see, but it's just showing that this is on the bottom here. This is just ferulic acid autofluorescing. It's just everywhere. So it just sits across the whole surface of the cellulose. Now, the important thing with that is that if you leave it for a little while and then you expose it to the conditions of the stomach and the small intestine, it sticks. It doesn't come back off. So this is a way in which not only that we can bind the polyphenols and related compounds to cellulose, but there is a prediction, and we're not the only ones saying this now, there's a number of people saying this, the prediction that they reach the colon. So we think this is another part of the story around whole foods, and we need to focus a lot more on what happens to micronutrients in the colon rather than worrying about whether they're bioaccessible or bioavailable in the small intestine, because the amounts that end up in the bloodstream are really tiny of these things, and somehow it's difficult to explain how they have such an effect. But if they're mediated through the microbiota, that's another story. So we think that's an important part of this as well. So I think I said earlier that bile salts was one area that was considered to be a known binding thing. So this is the beta-glucan story. So those of you who've seen the health claims around oat beta-glucan, reducing cholesterol, this is the proposed mechanism. It's proposed that the beta-glucan, we would say probably any viscous polysaccharide, but in this case the beta-glucan, can bind the bile salts, take it through the system, and because bile is actually similar to cholesterol, to make more bile, the body recruits cholesterol from the blood supply and thereby decreases the blood circulating cholesterol. So you can see there is a little bit of binding in vitro. It's pretty weak, but actually when you look in vivo, it may not be the important factor. So the one area where there was a good hypothesis, when we go and test it, we're not finding the evidence. We don't see that there's much bile that escapes digestion in the small intestine in pigs in our model system. So we have to sort of hold tight on that one a bit. But the other binding phenomena we talked about there are strong and really have to play a role in vivo as well as in vitro. So that's the binding. So what about transport? How do we modulate the way in which things move? So two ways. One is through the intactness of the cell walls as an encapsulating agent, and the second is as a viscous material, as a barrier to mixing of components. So intact cell walls, two examples at the extremes of the nature of the encapsulated material. So we'll talk about sugar as a very hydrophilic, completely water-soluble materials, and we'll talk about carotenoids as extremely lipophilic and water-insoluble materials. So we started looking at the release of sugar from cut tissue cubes, very simple experiments of apple, rock melon, raw carrot, and steamed beetroot. So this is just a set where we're looking at the percent that's released, 100% up there, under a simulated oral condition, mostly a simulated gastric condition, and then a bit of bile salt to kick in the rest of it. And you can see that there are somewhat different specific rates that are going on there, but for the individual, for sorbitol, fructose, glucose, and sucrose, the relative rates are the same. So that it's not a question, there's no difference between sorbitol and sucrose, so it's not a molecular size thing at that scale. But when you start looking at the sizes, so we're talking at cubes here of 3, 4, 5, 6 millimeters. So these are the types of sort of sizes you might think about swallowing. And here now the rate of release does make a difference. So that size, the difference between a 3 millimeter and a 6 millimeter cube, think about that. That's about the size that you would be swallowing. That does make a difference in terms of the rates. You can get about a third less sugar released over the first hour from a 6 millimeter cube compared to a 3 millimeter cube. And that really, it's simple, but it's a real effect. You can do a bit more scientifically, so you look at it in terms of rate coefficients and so on. So it's essentially a first order process, it's a diffusion process. And the sugar release rates, it's really a question of how many cell walls they have to get across. So we think that essentially smaller cell, it's not probably so much around differences between the cell walls of carrot, apple, melon, and beetroot. It's probably just the number of walls they need to get across, the number of membranes they need to passage. But these rates are sufficiently slow to influence the glycemic response. And when you look at the glycemic indices that are there for whole fruits, and you try and compare them with, well, we know what the fructose, sucrose, and glucose concentration is. So if we can estimate a predicted glycemic index, that estimated glycemic index is almost always higher than the measured one, and we think this is the reason, so that you can take your sugars in the form of a fruit and have less of a glycemic response than if you had the same amount of sugar but in a solution. So this is just to show you, I guess, at the extreme end, because glucose is about the most soluble component in our diet, and you'd have thought the most easily transportable, but still, even glucose is retained within cells sufficiently over the 1 to 2 hour time frame that it can affect our glycemic response. At the other extreme, this is just to show that a single cell wall is enough to prevent release of nutrients. So here are carotenoids. Nice that they're orange, so we can just see them directly within a carrot shred. And then if we recover pieces at the end of the small intestines, these are people who have had their colon removed, so that we can recover samples after processing through the stomach and the small intestine, but without a large intestine. What you can see is on the edges here, you can see that the carotenoids have been released from the edge of these materials, but you go one cell in and the carotenoids are all still there. So this is what it looks like after. This is a before and an after. They're essentially unchanged. So this and a number of other images, a single cell wall is enough to prevent carotenoid absorption in the small intestine. So carotenoids, most of them in intact foods, will be getting through to the large intestine. Carotenoids you eat in a pill, no, that's a different thing, because they're not restrained in this way. And again, we don't know yet. We don't know what the consequence of carotenoids mostly getting to the large intestine is. We need to understand these things. So intact cell walls are the barrier. So viscous digester also a barrier. So this is a bit more intuitive, so that if you have a thick solution, it's going to slow down the rate at which enzymes work. And I'll just flick through this, but the result is here basically that both the amylase digestion of starch and the transport of, well, we just use glucose, but the transport of the products of starch is not purely a viscous effect, because we can overcome it by efficient mixing. So if you mix well enough, in other words, it's a transport property, not a viscosity property. So viscosity and mixing together is what gives you the transport properties. I won't show that. I'll just flick to this one, which is these are the transport data. So we have viscosities here without any beta-glucan of 1, because that's water. And with 2% beta-glucan, we have a viscosity 100 times that. But if you go from unmixed in water and thoroughly mixed with 100 times the viscosity, you get the same mass transfer coefficient. So it's perhaps not surprising, but at least it shows that it's not just the viscosity, it's how it behaves. And so we need to understand what are the local shear rates and movement times in the small intestine, with the villi moving past each other and so on. There's nothing in the literature on this. I mean, people estimate and guess, but we really don't know. And this is where, in collaboration with engineers, they'll quickly tell you this is about the Reynolds number, which indeed it is, which is the way in which engineers look at things flowing through pipes. So it's about the density and the viscosity, but it's also about the stirring speed and the diameter of the stirrer. So and this fits beautifully. This is the Reynolds number, and it just collapses onto a single behavior for all of those different types. So they're behaving in a classic engineering way. It's just we tend not to think about that in the digestive tract, but we do have to do that. We have to understand what the equivalent of the stirring speed and the stirrer diameter are in the small intestine. How we're going to get to that, I don't really know, but that's a challenge. So finally, looking at the structuring aspects, and I'm gradually moving down the digestive tract here. So we're now entering the large intestine and thinking about what is it that causes differences in the rate of fermentation. Because not only we want fermentation, we want that fermentation to persist throughout the large intestine. So you probably wouldn't be surprised to learn that the rate of fermentation of cellulose is much slower than the rate of fermentation of some classic soluble polysaccharides. So that's a reasonable thing. They all have their characteristic growth curves against a particular inoculum. I mean, it will vary somewhat depending on what inoculum you use, but typically you've got a relatively small lag phase and a very rapid growth phase, reaching plateau around 24 hours, whereas for the cellulose, you're not reaching the plateau till about 72 to 80 hours. So much, and these sort of time frames are clearly relevant to the passage rate, our food passage rate of the order of 24 hours typically. So there's a big difference, as you might expect there. And I think, as I've said already, that fast rates are not always good. We want fermentation of carbohydrates because mostly the beneficial microbiota use them as their feedstock, but we do want that fermentation to continue towards the distal colon. So this work was really around some of our cell wall in vitro synthesis, where we find a way of making complexes between this cellulose and these components in a matrix which is quite similar to what happens in plants. And so the next slide will have this same picture here, but with composites of each of these three polymers with the cellulose. So if you keep that sort of picture in mind, when we make the composite, we do several things. First of all, the composites are all behaving rather similarly. Secondly, we've sort of got over this lag phase now, because we've now got some soluble material associated with the insoluble material. And it's not just a combination of the two factors. These are not just averages. There's something happening here. There's a cooperation between the two polymers that's happening. Not only are we getting this sort of continuous and smoothed out fermentation, this is a fermentation primarily of cellulose. If you look at some of these, this arabinoxylan composite is only 12% arabinoxylan, so it's 88% cellulose. So almost all of this is cellulose. And so the presence of something else on there, to a little sweetener on easy fermented material on the surface of cellulose, has a really big effect on the consequent fermentation of the cellulose underneath it. Another example is that it's not just the molecules that are there, it's how they're arranged architecturally that really has a major effect on the way in which they behave. So that's looking through those general properties. And now let's finish off just by saying, well, how can we take this forward? How can we construct some sort of framework that we can understand how things might work? And this is where we start thinking about what we currently do. And we currently have a terminology of soluble and insoluble dietary fibres. The way these are measured, you take your fibre materials, your plant food material, you add a lot of water to it, and you simply just measure what becomes soluble under certain conditions and what remains insoluble. And so you get that fractionation occurring. So traditional soluble fibres, things like pectins or guar gums or something like that, they're typically associated with viscosity effect. Beta-glucan would be in this, in the stomach and the small intestine, and fermentation in the large intestine. That's fine. Traditional insoluble materials, so brands and so on, are associated not so much with viscosity effects, but they are associated with increased passage rate, and that's something I haven't really talked about, but the effect of particulates on the way in which our body pushes digester through is an important factor as well. But also limited fermentation, so that they contribute to stool weight and regularity and so on. Those are fine for these sorts of materials. The issue is that almost everything we do eat, apart we should eat a lot of bran, obviously, but almost all the rest of the cell wall material we eat is intermediate. It's typically, they're often insoluble by phase separation criteria, so they don't fully dissolve. You might, more likely you get swelling. You take a lot of plant foods, you add water to it, you often get a bit of swelling. The things are trying to solubilize, but they're still restrained because they're still part of a matrix in the cell wall. So swollen and viscous in the stomach, and this is your porridge. This is how oats behave. Oats, it's not, you don't solubilize anything from oats, but you get that swollen feature that really gives that rheology in the bowl and also in the stomach and so on. But it's not because of solubilization, it's a swelling process. So it's really insoluble, but it's nothing like bran. So we have a problem here that it's not a good definition. Likewise, many soluble fibres are actually of quite low molecular weight, and they don't have these viscosity effects. So and the food industry loves these because they can pile them in. So oligosaccharides or inulins and so on, you can pile them in, and on the label you can say all sorts of dietary fibre contents. But you're only getting a small fraction of the potential functionality you could get. But it's because our labelling system at the moment just measures dietary fibre in a gram basis. And a trisaccharide is considered the same as a high polymer. Functionally, that's not true. Chemically, yeah, it's true. So another challenge is here how we're going to regulate or label in the future if we really want things that are more to do with functionality rather than composition. Similarly, there's also an assumption that solubility is related to fermentability. We've already shown that isn't the case, and I'll show you, I think, one example of that next. Yeah, so these are for cereal flowers, so endosperm flowers, that's white flowers, no brown. We've isolated the cell walls from these, and these are the same fermentation curve. You don't just see which ones they are. I'm just saying that they're all rather similar. The top one here are flowers, and some of them are the soluble fraction, some of them are the insoluble fraction. The bottom ones here are a mixture of breads and extruded products. So the extruded barley with all this beta-glucan was a very tough material, so that's the only one that's different. All the rest of them, though, essentially the same. So whether it's soluble or insoluble by that classic criterion is no use to us in determining how well it ferments. It's not a factor that's controlling. So what could we do? So we go back to these are the materials we're dealing with. We've got a whole range of different materials from relatively large pieces. So if we have a soft piece, you can swallow pieces of more than a centimetre quite easily if it's a soft tissue. So things of up to a centimetre is probably our length scale. And if you go down to things like the oligosaccharides, which are the minimum that you need to have a fermentation effect, they're down to the nanometre level. So you go from nanometre, 10 to the minus 9, to centimetre, 10 to the minus 2, 7 orders of magnitude in size. And it's always difficult to comprehend that level of that set of size ranges, but that's what we're dealing with, nanometre to centimetre. And of course, all of these types of structures contribute to all the physical properties I've been talking about. So they all have different behaviours in terms of their rheology, their molecular binding, and the way in which they deconstruct in the digestive tract. So size is one of those things. We have to take size into account, and size will have an effect on all those different properties in a way I'll talk about in the final slides. So we think that we should have a dimension which talks around the effective particle or molecular size at a certain point in the digestive tract. It's not in the food, it's what's happening in the digestive tract that's important. So you can, dissolved molecules, you measure their volume, so either with chromatography or with intrinsic viscosity. And for insoluble materials, you visualize, you assess their particle size. But that's not enough, because it's not just size. Clearly, the size of a particle is not enough to tell you what its properties are going to be. And so you need something around that, the density, the local density of that particle, as the second factor that really gives you some differentiation in terms of the properties which are likely to be nutritionally of value. So this is what we're going to do, and look at the properties of cell walls and dietary fibres in this two-dimensional plot. One axis will be size, and the other axis will be concentration or density. So as I said already, then we've got 7 orders of magnitude of size from nanometre to centimetre, and in terms of concentration, so we're talking about 100. Obviously, that's just all polysaccharides, so that's dry polysaccharide down to 0.1% solutions. But you'll notice both axes here are logarithmic. So the range of structures is massive, and our bodies are wonderful at coping with those range of structures. A range of structures in terms of sizes and also in terms of concentrations. We deal with them all across these many orders of magnitude in both dimensions. So here's just a qualitative, semi-quantitative, I guess, location of some classic types of food components. So if you've got cereal brands, which are relatively unhydrated, so they're nearly 100% polysaccharide. They're typically in the millimetre to centimetre level. If you go down to soluble polysaccharides, they're typically around the 10 nanometre size, and they're typically only in that sort of 1%. If you go above that, they start getting too viscous. So there are examples all the way through that. Clearly, if you have, if you've got oligosaccharides like inulin, they tend to be used at higher concentrations. So that's a typical usage area for inulin. And then if you think around this is for cereal bran, but you can have, or you could have, your oats would also fit into here. So fruit, vegetable pulps, things which are relatively large pieces, but they are quite swollen. So their polysaccharide density is only a few percent typically. So they're very much different to things like bran because there's all that water going through them. And then in between, we've now got, this is an area where a lot of the industry is trying to focus. So a lot of the new so-called fibres are really in this region. And they're typically things that are, for instance, recovered pulp from orange juice pressings, or one that we're working with from sugar beet bagasse. What are the fibres that you get? And they're typically either in the case of sugar beet bagasse, dense. In the case of orange or other fruit juice pulp ingredients, they're swollen cell wall fragments, but they fill this sort of gap in the micron sort of scale, where we can't perceive them individually, but they're definitely bigger than these individual molecules. So if we take that as a way of trying to categorize, what happens if we put this soluble versus insoluble criterion in there? Now, as I said before, the method for measuring solubility is you add a lot of excess water, and by saying that, that essentially takes this axis out of it, because whatever it was on that axis before, you're adding a lot more water, so it becomes less relevant. So to a first, and that's why these lines are fuzzy, because obviously this is not accurate, but it's just to give you an impression. So it's something like when a polymer becomes a particle, which happens in the hundreds of nanometers sort of scale, a little bit depending on the individual component. But there are very few molecules that are at the micron scale, and you don't really, particles at 100 nanometers are a bit difficult to define. So it's in that sort of range. So keep that in mind. That's where you divide. So I'm now going to do the same division for some very characteristic physical properties. So if we look at viscosity, first off, it's very different. Obviously, soluble, insoluble. So soluble, insoluble doesn't tell you anything about viscosity. And you've basically got two forms of viscosity. One is based on polymers. So obviously, the larger the polymer is and the more concentrated it is, then the more viscous you get. But at the other end, you've also got particulate rheology. And so when you get to things like brands, which have very little water absorption capacity, they're not contributing to rheology because they're just sedimenting. So you've got two very different mechanisms by which you generate non-viscosity or viscosity, and of course these are simplistic. Everything is a shade of grey here. I'm just putting it in black and white to overemphasize it. But as you can see, that doesn't really bear any resemblance to soluble versus insoluble. And when you start looking at what becomes slowly fermentable or rapidly fermentable, again, totally different again. And here, things that are slowly fermentable are either because they are very densely packed, and essentially that's because the bacteria now have to work from the outside in, and so it's a slow process. As soon as they get more open, so there's a very big difference between a cereal bran and a fruit or vegetable pulp. As soon as they get more open, like a fruit and vegetable pulp, the larger particles actually become more rapidly fermented, and that's because the bacteria can get inside the residual cells, and it's a nice ecosystem or nice environment for them to actually prosper in. So if you can get inside, it's a nice environment, and they prosper and they ferment more rapidly. If they can't get inside, like with brands, it's a slow process because it's an exterior etching type of process. And then the other reason why you can get slow fermentation is not really relevant in the digestive tract. This is just a water activity thing. If you have enough concentration of small molecules, that's an abiotic situation because of the water activity. But this is the one that really matters up here in terms of fermentability, and this boundary between controlling the fermentability from the cellulose or bran end through to the more hydrated cell walls is an area that we can play and understand a bit more. But again, different to viscosity, different to solubility. And then finally, talking about binding of things, it'll be a little bit depending on what you're binding, but in general, things will bind more to more rigid surfaces. So if you're a molecule and you want to bind to something, the entropy that you have to give up in binding is always reduced when you have a rigid surface to bind on, and that rigid surface also provides an opportunity for molecules to explore the landscape there and find the area that they want to bind to. And so in general, as you get to more solid-like surfaces, so as it's both larger and more concentrated, you will increase that binding potential. So I think the only point I wanted to show here was that if we believe that these sorts of physical properties are important in how things behave in the digestive tract, they don't have a single origin, that they all behave, they all map differently onto this sort of construct. So that's really what I'm going to conclude now and say. We know from the epidemiology that there's a key role for plant-based foods, and it's almost certainly down to the structuring of those foods rather than any particular micronutrient component, because the effects are particularly generic across a wide range of plant-based foods. There's a little bit of debate about the relative merits of cereal foods versus fruits and vegetables, but not between different fruit and vegetable types around the world which have very different micronutrient content. So it's pretty convincing that it's the tissue structure which is the core feature which controls the health value of plant-based foods. And that from our work, we look at these properties in three ways: look at them binding, transport, and structuring. The challenge, of course, is that we've been looking at things at one component at a time here. Obviously, that's not what happens in our digestive tract. There's a competition all the time. All these other components are going through at the same time. And so the trade-offs we also need to understand there of the multiple food components and the diverse and multiple digestive and nutritional outcomes. So in terms of how we're going to think that through, we want to make our 2D plot more quantitative and illustrate it with more specific things. I guess I see that we can use this to map the changes that happen to a single food through the digestive tract. How does it change its size and its degree of swelling as it goes through, and how can that help us understand the changes in its binding, transport, and structuring properties as a result? So that's the way we think about how we're going to use these things. That's on the one hand. On the other hand is saying in a single environment, what is the effect of different types of foodstuffs on the properties in that environment? So that's our anticipation, is that we want to map these properties so that we can make them a bit more generally applicable. That's it. So thank you to a whole range of people who I haven't acknowledged all the way through, but a lot of people have contributed to all of the data in here, and it's really, this is one of the core activities of the Plant Soluble's ARC Centre of Excellence, which is a very fruitful collaboration between ourselves and teams at the University of Melbourne and the University of Adelaide. And with that, I think I'll finish, and thank you for your attention.